Okay, I hope that's okay with you. Okay. All right. Um, anyway, so uh, actually, John and I met in a way that's sort of exemplary of what he's like scientifically in terms of being ready to think about anything and jump into some project and so on. I saw him out as a person who had an apparatus that could potentially discover a very, very small component of an exotic type isotope where something that other than a nucleon was attached to a nucleus. And he, it turns out, has just the best device for this thing, where you could be sensitive like a part in a billion. Um, and so it was very typical. I've now learned of him. I didn't know all these other things about his personality and things. And
He's a weirdo.
his response was that he's ready for anything. And so I just learned that he was involved in demonstrating, in fact, proving that the first natural quasi-crystal was actually a natural thing and not some sort of like fake or something like that. And the thing he is, I believe, perhaps most widely known for, I'm not sure you'll correct me, I hope, John, is for having developed methods, and I think he'll tell us about it in this talk. So his general area is taking advantage of isotope ratios to learn things about geochemistry. But along the way, it turns out isotope ratios, as he'll sure explain to us nicely, are sensitive to the temperature, and he can get insight into the body core temperature of dinosaurs this way. Anyway, if that's not a kind of come on to the audience, I can't imagine what would be. So anyway, thank you so much, John, for being here. I'm going to go sit there, I guess.
Well, thanks for the invitation, Glennis, and thanks for being such a great colleague. You know, that was basically our futile but ongoing hunt for evidence of sexaquark dark matter was the funnest thing I did in the lab during the pandemic. It was like my little passion project, and was really aghast. So let me see if I'm seeing myself and everybody else appropriately here. Yeah, so what are we going to talk about today? I'm not going to talk about dinosaur body temperatures. It's a fun subject. It's something I really enjoyed working on. If people want to talk about it later, that'd be great. Maybe when I come in the fall, we can do like a dinosaur body temperature seminar because it's a fun thing to talk about. I want to instead do something different, something that's more current in my work. Let me see if I can share my screen. Yeah, so I want to talk to you about some work that my group has been doing over the last 2 or 3 years, trying to learn something about life's origins on the Earth and to contribute to a big community-wide effort to look for evidence of life on other solar system bodies, or evidence of prebiotic, that is, chemical processes that could lead to life on other solar system bodies. And this is a subject, I'm sure it's not particularly close to any of your areas of research and expertise, but it's something that really all of us are part of in a way. There's such a big international effort behind space missions to go to Mars, go to outer solar system bodies and so forth, motivated by this question that basically anybody who's a scientist, anybody who reads a newspaper, you have an on-ramp to this subject. We're all sort of engaged. And also what I find interesting about it, really the reason I got involved in this area of research, is it's one of the few things you can work on that's genuinely scary. It's like almost not science. Because the way that, you know, the search for life's origins, who can say, you know, where's the evidence? What do you even study? There's a lot of meta science hovering around this question. And so I took it as not exactly a personal challenge, but I was interested to see, is there a way that we can find something in this that is science and that you can somehow move forward with it? So that's our main subject. There's also a subtext here, and that is that what my group specializes in is developing new ways of observing the natural distribution of the naturally occurring isotopes. So like deuterium and carbon-13 and oxygen-18 and things like that. Really seeing their distribution in new ways and in ways that open up the complexity of the questions that you can ask about natural materials based upon their isotopic contents. So there's a bit of technology and chemical physics and things like that slid in a sneaky way to this lecture. And if you enjoy it, great. If you don't worry about it. You can just focus on the space science part. And a whole bunch of students and postdocs have helped, and a bunch of people have paid for this. You can see them listed there. So, okay, so here's a very recent event that probably all of us have read about that is one of the motivations for the work I'm going to show you. It's the Mars 2020 landing, the Perseverance rover, which I have to say, one of the more unfortunate names for a spacecraft. Like, you're putting up with having to do this mission. You're somehow persevering. Like, aren't you excited to do this? Can't you have a better name for your rover than that? But that's what it's called. It's a Perseverance rover. And this is a thing that landed 2 years ago, almost to the day. It was like back in late February, in the Jezero crater, which is an ancient impact crater on the surface of Mars that was a lake at some point in the distant past, fed by a river that you can see feeding in through this river channel, creating a delta, sort of like the Mississippi River Delta. If New Orleans could sit right there, and this would be the Gulf Coast, there would be the Mississippi River Delta. And so we landed a robot there and started driving around. And one of the things that we did was to equip this robot with mechanical devices that could abrade patches of rocks that are found on the surface of Mars and measure their chemical constituents. And then a second device that could drill deep holes, like 6-inch deep holes. This is looking down the bore of that device, and then extract tubes of rock that are sort of like the diameter of a piece of chalk and maybe 4 or 5 inches long, something like that. So there are dozens of these things that are getting sampled and being brought back to the Earth for the study in terrestrial laboratories. In fact, I'm guessing either me, or if I'm too old to do it, somebody like me will study these things in the way that my lab does. So why are we doing this? This is billions of dollars, decades of effort, all kinds of different countries, hundreds of scientists are involved in this. Why are we doing this? And there are a lot of different motivations that each individual brings, but the only real collective motivation that justifies the whole activity is to seek for signs of ancient life, either life or chemistry that could occur on planetary surfaces that we imagine could have led to life on the Earth. And the issue here is the Earth is so dynamic geologically that evidence of its early history, particularly ephemeral things like organic molecules, very difficult to hunt down. Mars isn't really geologically that active. And so you can find objects that are billions of years old and maybe only modestly changed from their original condition. So this is the motivation. There's another mission almost exactly at the same time going on in the outer solar, well, this one's in not that far, but it's an object that samples the outer solar system. This is a mission, the OSIRIS-REx mission, that went to the surface of the asteroid Bennu and touched down and collected some rocks. And this is a sort of complementary, like a flip side of the same motivation. Here, the idea is to collect a very primitive object that has not undergone planetary differentiation or geological history, and to look at organic constituents of that rock as sort of original precursors, the materials of organic chemistry that eventually planetary processes could use as substrates for life to emerge. So we're looking for molecular precursors for the origin of life. So both of these missions, they're just the most recent and most visible instances of a now 20 to 25 year long concerted push to look for evidence of life in the solar system, to look for evidence of the emergence of life in the oldest rocks on the Earth, with a few really first order questions that have to be dealt with if you're ever going to succeed at this. One of them is, can you tell the products of life, if you ever find them on the surface of Mars, or we go to some outer solar system body like Enceladus, where we see organic matter, can we ever tell that we're looking at the products of life as opposed to some non-biological process? Second, if you find, whether you find evidence of life or evidence of prebiotic chemistry, that is like not life, can you tell how exactly it was made? Can you learn anything from it in detail, or can you just recognize that it's there? And then a third sort of related question is, can we figure out the conditions of environments where life could have emerged and existed? Like, we might discover evidence of prebiotic chemistry in one place, and by recognizing the conditions where it occurred, then we have a revised estimate, a revised understanding of how prebiotic chemistry would have worked in the early Earth. Okay, so that's the basic motivation, the basic goal of all of this mission work. How are people doing it? What's the sort of guiding intellectual infrastructure behind this search? And it can be divided roughly into two different approaches. There's a top-down approach where you're using telescopes, landers, anything you can do to try and find instances other than in the modern Earth where life is active. And this might be a search for plants and animals. You might get like super lucky and we find an eyeball rolling around on Mars or something. More likely, we're looking for single-celled organisms. People often rationalize this. They'll say like, oh, I'm looking for single-celled organisms because it's obviously the best way for life to be on some planet other than the Earth. No, they're looking for single-celled organisms because we already looked for things like trees and we didn't see any. So this is like by default, all that we imagine. It's the best we can hope for. So this is the top-down approach. Basically, you're swinging for the fences, hoping for a home run that you discover life in action or fossil remains of life, maybe. The other approach is bottom up, where you say, you know, I know enough about the universe and about planets to understand that organic chemistry is sort of ubiquitous. And somehow it organized itself in such a fashion that it created simple organic molecules, formaldehyde and methanol and things like that. Somehow those things formed amino acids and nucleobases that are absolutely essential to life as we understand it. Somehow those things oligomerized or joined to each other and created things like sugars and lipids and things like that, that organisms absolutely have to create metabolically to live. And then something joins these two worldviews together. Okay, somehow we made this jump from the most complex organic molecules we can see nature making to the simplest life forms that we know about. Something is in between these two that we will try to figure out. And because nobody really has a good idea about how you will just intuit the way that these two parts of this problem join, we're hoping that just by working our way towards them, towards the middle, we will get some better ideas, that we'll learn so much about the bottom-up approach and so much about the top-down approach that we will eventually figure out how they should meet up. Okay, so what is the specific evidence? What are people hoping that they will find on the surface of Mars? And this is a pretty good example of something. If you're ever going to open the New York Times and you'd be like, wow, they couldn't even get it all above the fold. They needed the whole front page to tell me about this, the discovery of life on Mars. It'll be something like this little dark squiggle that you see here. This is the oldest evidence of microbial life on the Earth. And I put a little question mark by evidence because, you know, you need to look at it with the eye of love to really see it as alive. So this is a carriginous material. That means it's like a macromolecular organic material, a little bit like graphite, but less crystalline, less ordered, more junked up with other elements, nitrogen and sulfur and so forth. But it's closer to graphite than it is to anything else that you regularly encounter. And it just happens to have a shape that somebody could draw a cartoon of that reminds them of a multicellular organism. You will be forgiven for thinking this is not enough. And maybe it isn't enough, but it's the best that we can do in rocks of great antiquity on the Earth, meaning like 3.5 billion years old. And it's the best that we've done so far. So this is the kind of thing people are hoping that they will find, objects that are made of carbon and have shapes that kind of remind us of living things. The problem, of course, I mean, you'll immediately intuit, is what if it's something that has been called a sport of nature, meaning nature somehow conspired without biology to create a thing that looks like life. And this is what a lot of people thought fossils were when they were first being studied in the 17 and 1800s. Here's a perfect example of a sport of nature that ended up being very influential in the modern era of the search for life on other planets. These are some figures from the top are from a figure by Dave McKay and a variety of other colleagues published about 25 years ago that purported to find evidence, clear evidence to them of life on Mars. And this is a meteorite called Allen Hills or ALH 84001 meteorite. How we know it comes from Mars is a whole other topic. It's fun to discuss, but we don't really have time to get all the way into it. Let's just say we're confident it comes from Mars and it's very, very old. In it are a bunch of things. There's some organic carbon of ambiguous nature. There are some minerals that grew precipitated from water. And there are these little bumps, these little boogers, okay? So even looked at with the eye of love, these just look like little bumps. Nobody is really terribly compelled that these are cellular organisms or their fossil remains. Even the people who think that this is compelling don't think that this is compelling. And so this is sort of the line that we're at with this approach is how does it make you feel when you look at a shape of an object like this? Is it enough or is it not enough? You can get a sense of how difficult of a problem this is. There's a second line of inquiry that the top-down approach uses that is much more grounded, let's say. And that is the search for biomolecules, organic molecules that are so complex in their structure that it's difficult for us to imagine that they were synthesized by non-biological means. And there's a bunch of these that you might imagine we would look for, like maybe DNA, that would be great. Or how about proteins? Those seem like very complicated biomolecules that nature would never make by accident. Those would be great, except they're very fragile in natural environments after their formation. So they just degrade almost instantaneously to materials that are then unrecognizable. These classes of compounds I'm showing here, the steranes, where this little funny cartoon, if you're not used to it, each corner of these funny shapes is where a carbon atom lives, and this is just like a skeleton of the links between all of the carbon atoms. Here are steranes. These are the somewhat geologically processed versions of sterols, which include things like steroids, like the hormones in your body. When you die and all of your organic molecules break down, the hormones in your body will end up looking like this. Okay, and then there's a microbial version of this called hopanes, which is the same kind of idea. They don't use them as signaling molecules, they use them as structural components of their cell walls. But we can recognize these things, and they're very resistant. They're tough to break down, and so they persist in the environment. And even though you could make them non-biologically with enough work in a wet lab, a good chemist could make these things. It's not very plausible that nature would make them without life, without enzymatic processes to shape their assembly. And so these are considered very definitive biomolecules, and these would also be considered proof of life. You'd also read about this on the front page of the New York Times if they find it on Mars. Here is the problem, though, so far. So there's another rover that landed on Mars a decade ago called the Curiosity rover. That's a much better name. Better to be curious than just to be persevering. And it included in it a mass spectrometer and a variety of other complicated devices that were looking for, really they were looking for evidence of the nature of the environment at the surface, like was water there, was it a lake, things like this. But it had the capacity to detect organic molecules, and so they looked. And here is a chromatogram, meaning it's a time series of release of molecules from a chromatographic column in an experiment where they take a rock and they heat it up They boil off all the organic molecules. They pass them through a chromatographic column so they become separated from each other based on their chemical structures. And then you observe them in time as a time sequence and see what they are. And so a bunch of things came out that loosely remind us of the chemical structures that we would like to see, these steranes and hopanes and things like them. But you know, if they are pieces of that, they're really, really, really degraded. There's nothing left but individual rings that have had sulfur atoms substituted into them, which is how you vulcanize rubber. So basically, if this was the remains of life on Mars, something cooked them so profoundly and chemically reacted it so thoroughly that now you just have vulcanized rubber left in little teeny bits. And so the question is, what do you do with things like this? How do you learn anything about it if you have so profoundly changed its chemical nature? This is a subject called taphonomy. Taphonomy refers to the preservation and really the transformation into something that is fragile into something that can be preserved. Fossilization of like a bone, that's a form of taphonomy. Molecular taphonomy is getting a labile molecule, something that wants to break down, and condensing it, curling it up, getting rid of some easily reacted molecules, getting it into a form that it can live for a long time in the geological record. So this top-down approach, even if it succeeds, it will not be enough for us to say that we have learned something profound about the origin of life. And that is because it will only see so deep. Okay, if you start at the top and dig down into the problem of life, you're only going to dig as far as you find evidence of recently living things. And so if we dig down to the bottom of the Earth's tree of life, we find this thing that's referred to as LUCA, the last universal common ancestor. But of course, before that, we're all of its ancestors. This thing didn't just like spontaneously spring out of a bunch of dissolved organic molecules in water. A whole series of processes, living and pre-living, conspired to create LUCA, even if we found it. Okay, and by analogy, imagine that you are like a car aficionado. I don't really like cars, but other people like cars. So this is a useful metaphor if you're one of those people. Okay, imagine you were trying to figure out what was the earliest car like just by looking at traits of existing cars. You might get something close to this goofy looking car from the 1800s, but you would never by that approach figure out that this thing used to be a horse, like that used to be that we would ride horses instead of these funny looking cars. So it's a similar kind of problem that you encounter. The other side to this problem then, the bottom-up approach, what is their organizing idea? How are they going to try to make progress? And I'm one of them, so I care about this. So first of all, you have to decide what will be the characteristics of a chemical system that you say is alive. And you have to be specific about this, but also open-minded enough that you'll give yourself a chance to discover something that isn't identical to things you already know. So this is the definition people have settled on, that life is self-sustaining chemical system. So it has to have probably some kind of cyclical chemistries that are like our metabolisms, where you can feed something in and at steady state do a bunch of transformations and spit something out the other end without it just being a unidirectional process that goes blah. And it, like cooking, you know, when I fry an egg, that's not a steady state process. You fry it, it stays cooked, and that's it. So you need some sort of sustainable process. It needs to be capable of Darwinian evolution. So what's needed here? We need to be able to support chemical reactions, drawing on the environment. So we need to have something that's like a network of chemical reactions. It's a system that is different from just the rest of the universe. So I need to be able to draw boundaries around this thing I'm saying is my living system. So it needs to somehow be able to encapsulate itself so that I'm different from the air in the room. And it needs to be able to undergo some sort of evolution, which means I have to have a chemical mechanism of inheritance so that if I duplicate myself or I grow, somehow the information about what I am gets carried forward. And yet, if I'm going to undergo Darwinian evolution, I need to be able to have variation or mutation of this way that I store information. And then as long as I have those two things, nature will take care of the rest by selecting the weaklings and saving the ones that are good at being a chemical system. Okay, so let's be more specific about what exactly is called for. We need to somehow have structural confinement that life we recognize does as cell walls. And people have worked on this for a number of years and they haven't really thought of anything better than using various common organic precursors to make fatty acids, which we'll see some examples of in a minute, which can then be combined to make lipid structures, and that makes cell walls that will keep the environment out and your goop that you call you in. You also need information or replication molecules, and there are things that are like RNA, like DNA, nucleic acids that are unfamiliar to us and that life doesn't use that are possible. But RNA and DNA, they're probably the best bets. They're the best guesses about how this information molecule would work. And it's because the same precursors that we know are in the environment can make nucleobases, which we'll see some examples of, and those can spontaneously oligomerize, stick to each other to create self-replicating molecules. So we kind of see a path of non-living chemistry to this function. We then need to be able to use precursors to do metabolic, like energy transfer chemistry, things involving sugars. We need to have some way of storing and using energy, shuttling energy. So that involves complicated organic molecules. And then we need some way to control kinetics. So our bodies control kinetics with enzymes that are basically proteins. RNA can also be an enzyme. So there's other ways you can do enzymatic activity, controlling the rates of reactions. But we need to be able to do this so that the reactions that are useful can occur, while reactions that are not useful can be inhibited. Okay. So this is all kind of complicated. Like, how are we supposed to solve all of these problems at once? The discipline that studies prebiotic chemistry and the emergence of life, they have sort of split themselves into two camps. There's a camp that tasks itself with figuring out how we go from ubiquitous substrates that we find all over the place in the universe to what you might call enabling organics. Organic molecules that we know we can figure out ways to form without biology, but biology absolutely needs them. And we can't think of any forms of biology that don't need them. And then the second half is taking those enabling organics and really organizing them into larger oligomers, big interconnected structures that do critical functions like RNA, like cell walls, things like that. So this second part, the oligomerization, I'm a geologist by training and a geochemist. I like things that are preserved in rocks, okay? Things that I can study and make inductive inferences about. I don't like going into the wet lab and playing make-believe and seeing what I can make happen in the lab and then imagining, well, what if it had happened somewhere else? This doesn't interest me. I want to be able to learn something from an object from nature. And unfortunately, these oligomerization, organization sorts of chemistries, these are the things that are easiest to degrade and they kind of fall apart very quickly. Maybe someday people like me will figure out how to study them, but a better target to me is to work on this first step. How does nature really do this chemistry? How does it launch itself on this first step towards life? Okay, and so this is what we're working on. So the very first steps will be familiar to any astronomer or anybody who follows astronomy. You just look up into the sky with some sort of rotational spectroscopy or vibrational spectroscopy, and you quickly realize that the chemistry of the universe is basically organic. And there are a lot of interesting starting places as substrates for organic chemistry. Methanol, and water, of course, and cyanide, very useful, formaldehyde. There's a lot of interesting little molecules that get synthesized. Many of them get made in very cold environments by grain surface chemistry. There's a certain amount of vapor phase chemistry that happens that precedes this, but the chemistry that leads to most of the things that catch our attention spectroscopically, probably they happen on solid surfaces of water ice grains that get irradiated. That creates excited state little chemical microenvironments, which are then highly reactive and lead to the formation of carbon-carbon bonds and carbon-nitrogen bonds and other sorts of chemistries that are difficult to do in the vapor phase when it's cold. A second environment are stellar envelopes of cool carbon-rich stars where you will just condense out basically soot, polycyclic aromatic hydrocarbon sorts of materials. And we think both of these chemistries are, first of all, they're ubiquitous and they're plausibly the factories that make the starting constituents that we could work with. We also see by spectroscopic observations of disks around young stars that certain things that are interesting to us, like methyl cyanide is another of these simple precursors, can be seen forming in excesses beyond what you'd see in a nebular environment around disks. So presumably there's some sort of chemical processing that's occurring in disks. And then if we look at comets, very primitive objects from our outer solar system, there was a mission sent to a comet that did relatively sophisticated mass spectrometry on the gas and dust that blew off the surface of it. And they found amino acids. They found at least an amino acid, glycine, which is the smallest, simplest amino acid, and phosphorus was detected in a potentially usable chemical form. So this builds the story that the universe is just in the background, spontaneously creating things of use to our story. But so far, nothing approaching the full suite of enabling organics that we need has been found in these environments. We don't have dozens of amino acids that have been found in these settings. There are no nucleobases. There are no fatty acids. Like, we're not finding the full suite of things that we need. And so there's this sense that if you're going to create a chemistry of life, you must do it in a planetary or maybe asteroidal interior, some sort of environment where you have time, liquid water, mineral surfaces as reactive surfaces. You need some setting that is just not being encountered in interstellar space, nebulae, disks, things like this. So here's an observation, relatively recent, that suggests even a primitive planetary body can get you a long way that you are not getting off of planetary bodies. So Enceladus, an icy moon of Saturn, was visited by the Cassini spacecraft, and it did a mass spectrometric measurement of the dust and gas that was blowing in vents, jets that are coming out of the south polar region of Enceladus. And the mass spectrometer that they used is it's basically garbage, but it's the best that you could fly. And these little bumps and wiggles, if you really are forced to interpret them, you would interpret as a family of organic molecules that are much more complex and much larger in molecular size than things that we're seeing free swimming out in space. And so the thought is maybe the interior of this is a place where you could synthesize organics that are needed for life. Okay, what are the mechanisms that people think about? So one of the most famous early specific chemical mechanisms people looked at is the Miller-Urey experiment, where you basically take a reducing atmosphere, do electrical discharge in it. The electrical discharge in an environment with ammonia, nitrogen, carbon monoxide, and so forth will create cyanide, which ends up being a really critical reactive intermediate. And then that allows you to synthesize amino acids. So out of this came one of the earliest ideas about prebiotic synthesis, that this sort of chemistry, the Miller-Urey chemistry, was happening in an atmosphere. Its products were falling into an ocean, and then that would form a kind of organic soup where you could do whatever the next stages would be. This was quickly realized that when you start putting numbers on this argument, it would be way too dilute, that there's just no way for molecules to find each other to do any further chemical reactions. So this has sort of fallen out of favor. And anyway, there's no atmosphere on Enceladus, so that's obviously not what happened there. Second idea that's very popular right now, I would say it's the most popular concept for how prebiotic chemistry occurs in planetary environments, is if you have a structural rift, a crack really, in the crust of a planet, and there is magmatic processes, so ascent of silicate melt beneath that crack, and there's water present, say an ocean or water in pore spaces in the crust, you'll set up systems of circulating fluid. And those fluids will vent. When they get hot, they ascend, and they will vent into an overlying water body. And where they vent, they will undergo chemical reactions where their dissolved constituents can crash out of solution when they quench and cool. And they'll typically create sort of pipes of minerals. They get sort of encrusted in place. And the walls of those pipes present a setting that reminds some people of the metabolic situation that cells try to create between the inside of a cell and the outside of a cell. Inside is something alkaline and a little reducing. Outside is something acidic and a little oxidizing. And this is an environment where you can imagine doing redox chemistry that involves transfer of electrons. And electron transport is really the essential task of metabolic chemistry, is harvesting electrons, moving electrons, depositing electrons, and doing this in a controlled way so that energy, chemical energy, can be generated. So this is a setting that many people find very attractive. The trouble is, if you go there, there's no prebiotic chemistry really happening there. And when you try to do it in the lab, you don't make much. So other people don't find it very compelling. There's another argument about this kind of chemistry, the chemistry that happens in these hydrothermal vents, and that is it's a little too simple. You can do a little reduction, do a little oxidation. Oh, by the way, somebody is not on mute. I'm not on mute, but I'm not supposed to be on mute. If you're not on mute and you're supposed to be, maybe do that. So another problem is the chemistry that enables life is it's one thing it's characterized by is a tremendous complexity of networks of reactions. That compounds don't simply get made and then die. They get made and then used to make something else, which makes another thing, and that begats that, and that begat that, and that begat that, and now you're back to the beginning. Cycles of chemistry. So here, for example, is the central metabolic network of E. coli. And the only thing you're supposed to take from this is it's complicated, right? That there's lots and lots of nodes, meaning lots and lots of compounds, lots of lines, meaning lots of reactions. So it feels like if we're going to do prebiotic chemistry, we need to be able to have all of these compounds of interest interacting with each other complexly. And this has inspired another model. I would say it's the last one I'll mention to you, but it is in competition with, in intellectual dialectic argument with the hydrothermal vent idea. And this is a concept that life emerged in surface lake environments where sunlight could do some initial excited state chemistry, which then allows ubiquitous substrates like cyanide and carbon monoxide and things like that to form simple organic molecules. But then you go through cycles of evaporation and dry down, maybe impact and heating or volcanic processes. You cycle these compounds through different environmental conditions. And in each different environmental condition, they'll undergo a different family of reactions. And if you have enough time and you do this enough times, you will end up being able to kind of brew up the mix of things that we need. And this is called the cyanosulfidic hypothesis. And as a laboratory model, if I go into a hood, definitely use a hood because all the chemicals are poisonous, and cook up amino acids, these AAs, sugars, lipids, things that your cell walls are made of, nucleobases, which your RNA and DNA need. All these things can be created under a family of related conditions where I can do a little bit of the chemistry over here, and then maybe I pause and turn the temperature up and do a little bit of the other chemistry over here. So long as I can do this all nearby each other, maybe it all can come together into a working network. So really what we want is not a single reaction, we want a network. Okay, now we come to what my group is able to contribute to this. How are we supposed to tell the difference among all of these crazy ideas? We have things happening in space. We have vents. We have lakes. We have atmospheric things. We have the ocean. We have life, maybe. How can we tell all of them apart, particularly if they're not mutually exclusive? So it could be that all of them are contributing in some fashion to a sample that you find. So the way that my group approaches this is through the use of the distribution of the naturally occurring isotopes. And you're all sophisticates, so you understand what isotopes are. But the ones that we particularly care about are not radioactive nuclides or nuclides created by spallation or other sorts of energetic processes. We're interested in the stable isotopes that are rare forms of common elements. So for example, for carbon, carbon-12 is obviously the common version. 1% of it is carbon-13, and it's not radioactive. It doesn't do anything. Nothing makes it, nothing common makes it by radioactive decay. It's just there. Carbon-14 undergoes decay, and I'm less interested in it. Carbon-13, it's exactly like all the other carbons. That's what the word isotope means. Isotope, same place in the periodic table. So it has the same electronic structure, the same chemistry, almost. It almost has the same chemistry. The difference in nuclear mass changes vibrational energies of the bonds that it's in, and therefore free energies of formation of the compounds that contain carbon-13. And because of the depth of the vibrational energy wells, the activation energies for irreversible reactions. So a little bit of carbon-13, it makes the molecules different. It's like a little bit different molecule. So these sorts of differences in chemical behavior lead to so-called isotope effects. And one of the most famous of them for hunting for evidence of life in the geological record is the biological fractionation of carbon-13 from carbon-12 by metabolic processes. So for example, carbon-13 is about 1% of carbon-12, but if I take CO2 in air and fix it by photosynthesis in a leafy plant, then I deplete the carbon-13 in the product organic matter. And I do it for a variety of reasons, actually. It's not a simple reason. I do it because the CO2 has to diffuse into the leaf, and that's isotopically fractionating. And then there's an enzymatic activity that fixes the CO2, and that has a chemical isotope effect on it. And the net effect depletes by a few percent relative how much carbon-13 was there. If I change some of the details, like I use a grassy plant that has a slightly different mechanism of carbon fixation, I'll have a little bit different separation. If I am a microbe in somebody's gut and I take pre-existing organic matter and then say do methanogenesis as my metabolism, I might like double fractionate. I take a thing that was fractionated and I fractionate it again. So you can see we make this diversity of carbon isotope contents in the products of life. And this is something that people hunt for as a signature of life. This is probably the most widespread, frequently cited evidence on the Earth of the activity of life during the earliest periods in the geological record, which is called the Archean eon, like 3.5 billion years ago. And what's shown here is a depletion, a lower value of carbon-13. The units here don't matter. It's a funny little delta thing and a per mil. Don't worry about that. Lower means less, higher means more. That's all you need to know. There is less carbon-13 in organic matter than there is in inorganic carbon from the same era in the same sorts of environments as sampled by carbonate minerals. And that difference reminds us of the photosynthetic carbon fixation, ergo, it's evidence of life. The problem is there's a lot of different chemistries that reduce carbon. And we see them in photosynthesis, we see them in microbial metabolisms, we see them in petroleum systems where we take pre-existing organic matter and do chemistry to it. We see it in meteorites, we see it in the Fischer-Tropsch chemistry, which is an industrial chemistry used to make synthetic oil. You see it in lots of different places. This depletion in carbon-13, it's not really a characteristic of life, it's a characteristic of chemistry. And so if we just have this brute force measurement of how many carbon-13 atoms end up in organic matter, we're never going to figure this out. All of the different processes fractionate the isotopes of carbon. So how are we going to solve this? And the way that my group has approached it is to develop a new way of studying the isotopes of carbon and other elements, hydrogen, nitrogen, everything really is open for negotiation, if you will. Where we try and get more information. And by more, I mean a lot more information about molecular origins. So one way that you could measure isotopes in something is instead of as a bulk material like old fossil carbon, what if I could study individual molecules? Okay, people figured out how to do that decades ago, and it was very helpful. What if I could see differences between different atomic positions in isotopic content? That's even better. Now I'm tracing individual atoms that maybe trace individual chemical processes. There's also versions of molecules. In fact, most unique versions of molecules have more than one rare isotope in them. What if I had a carbon-13 and a deuterium in the same molecule and they were bonded to each other? That's a different version. What if I had 2 rare isotopes or 3 rare isotopes or 4 rare isotopes and they were spread around the molecule? Those are different versions. In all, and I can do this for all the different elements, so in all, there's over 4,000 isotopic versions of this simple organic molecule, alanine, which is an amino acid. And there's probably 100 of them that we can measure. In fact, there's a molecule not so different from this that somebody in my lab measured 200 isotopic versions of. So there's many, many, many isotopic versions. And the key insight is all of them are unique. They all have unique free energies of formation. They all are unique in their chemical reactivities. They're all unique in their probabilities of forming by different chemical reaction pathways. Each of them is a separate vote on how a molecule formed. So there's the processes that we might want to study are inheritance, the transfer of an atom from a precursor to a product; chemical kinetics, the fact that when I transfer atoms from a precursor to a product, there might be isotope effects on rate, like I was talking about a moment ago. And then there are reversible equilibria as well. A lot of the chemistry of life is actually reversible. And then there's transport processes where isotopically light forms move faster than isotopically heavy forms. So there's an analogy between what we're trying to do and a familiar field of science, genomics. So everybody knows that the genome, through its structural complexity, has, and its function and its ability to mutate and have inheritance, is a tremendously important chemical entity. But we're only really able to do something with that because we are able to observe the structural form of individual samples of DNA. And through that technological capability, that then enables all the things that we think of as being modern genomics, the forensics and the diagnosis of disease and all that stuff. All of it is because we are able to take one sample of DNA and describe its structure. So a big goal of my group is to create the enabling technologies that let us observe the isotopic structure of everybody else. So DNA is special because its chemical structure is so unique. Most organic molecules are kind of anonymous. You know, one alanine molecule is the same as another, but their isotopic structures maybe are not. And so that's the idea. So the way that we do this, I realize we're getting close to the end of the hour, so I'll just like step on the gas and accelerate through this. So the way that we do this is through mass spectrometry, but we do it in a very specialized way. And the basic concept is if I take a molecule and ionize it in an energetic way, it will spontaneously break into an idiosyncratic pattern of fragments. Each of those fragments samples a different portion of the molecule. And if I were to go then to one of the families of peaks that are created from one of these fragments, I would see a version with no rare isotopes, and I would see a big peak that has 1 heavy rare isotope and another peak that has 2 heavy rare isotopes, and so forth. If I zoom in further and I observe them with high enough mass resolution, I would realize that because the mass anomalies, the mass defects of all the different isotopes are unique, that is, carbon-13 is not 1 AMU more than carbon-12, it's 1.003 something, something, something. Deuterium is not 1 AMU higher than hydrogen, it's 1.007, blah, blah, blah. They're all unique. And so if you measure them with sufficient precision in their mass, every one of them becomes its own observation. Okay? And so if I can do this for every fragment, think of all of the information I can harvest about the isotopic structure. So that's the basic idea. The way we do this is through Fourier transform mass spectrometry, which is a very exotic form of mass spectrometry. Unless you've done it, you probably don't know very much about it. But the basic concept is that you take your population of ionized versions of a molecule and you put them in a cavity that's confined electromagnetically so that a charged particle can't really escape. It can just rotate harmonically inside of it. And you excite them into a motion at some frequency range that you feel like you can control, usually like radio frequency range. And then you observe them sort of remotely almost like listening with an antenna. You observe them as an induction current that is created in some conductor whenever a charged particle sweeps past it. That leads to a time varying signal as these packets of ions sweep past the detector. And if you do a fast Fourier transform on that signal, you'll see the fundamental modes are the individual frequencies, each of which corresponds to a mass. So that's how it's done. And the reason we do it is it has unbelievable mass resolution. So every one of these peaks pops out. The disadvantage is it's like not considered a quantitative analytical tool. So that was my group's contribution, is to make this be a quantitative analytical tool. So one version of this, you can go to a national lab and go to a device the size of a cement truck, literally, where the confinement of the ions is through a huge magnet that's liquid helium cooled. And you can see some illustrative peaks of isotopic versions of big organic molecules. In my lab, we do the same thing, but with a little teeny device the size of your thumb, where the confinement is electrostatic. And it's called an Orbitrap. And it's just a very, 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 very carefully designed ion trap. Okay. So we have like literally 5 minutes. So I'll just like race through. Well, what did we learn? So this set of techniques, studying isotopic structure to reconstruct synthesis pathways, we've approached the organic compounds in primitive meteorites. And these are most like the samples that are being brought back from Bennu by these asteroid sample return missions. And they're full of organic compounds, everything you can possibly imagine. So here's an illustrative finding of what we see when we measure the isotopic structures of these things. So here's the amino acid alanine. And by using these methods, we showed that the carbon-13 content is tremendously heterogeneous within it. That this central carbon atom that's bonded to nitrogen is enriched by over 100 per mil, so more than 10%, relative to normal terrestrial carbon, or the other carbon positions in the same molecule that was sampled. This is like 5 times the strength of chemical isotope effects and is a signature that something very special was inherited by that one site of this one molecule. So the next step to our procedure is then we go in the wet lab and we hypothesize a mechanism by which that molecule could have been formed from precursors, and then we document the isotope effects of that on every site. This is like a little second order correction that lets us say, okay, we spotted something special. Now let's hypothesize a chemical mechanism that will tell us what its precursor must have been like. So that leads to a model like this, where we imagine that there's some precursor. We've decided it's like a carbon monoxide that's rich in C13, because that's something that can be seen in astronomical observations, that gets inherited by one atomic site of a precursor aldehyde and carried through this cyanide-rich chemistry to make an amino acid. Okay, and then we fill out all the rest of the information with our experiments. We've done this half a dozen different times with different related molecules. And then we've leveraged our interpretations of these patterns of isotopic substitution by trying to interpret the molecule average data, which is a huge amount in the literature, through the same sort of interpretive framework. And what basically the method is, we'll say, if my precursor looks like this, and I go through the following chemical steps, then all these other molecules that I haven't had time to look at yet should have molecule average isotopic contents like so. And the predictions are all the little circles. And then the data are the things that aren't circles. So we're basically pattern matching to see, is our hypothesis extendable to other classes of compounds? And we do it with the aldehydes, and we do it with the amines. And the end result is this reaction network that says, here is our version of the KEGG network that we think operated on an asteroid. And it allowed very simple things that we know are ubiquitous in the universe, like carbon monoxide and hydrogen and cyanide. They reacted in these specific ways in order to create, in the same time and same place, this brew of amino acids and nucleobases and fatty acids and all the rest of it. All the things basically that life needs got made in this place by this mechanism. So the model that we put together most looks like the cyanosulfidic hypothesis. It's obviously a different setting. An asteroid is not a lake. It's more like the pore spaces in an asteroid are doing chemistry that resembles what the people talking about lakes are talking about. So it's a sort of natural model for what they mean. Okay, and if it's like any one natural setting that's important to us, it might be like Enceladus, like the interior of Enceladus. Okay, so we're basically out of time. The last thought I want to leave you with is this is now a model for how we might approach the study of really juicy, interesting organic compounds that might come back from Mars, things like amino acids. We could do exactly what I just showed you and maybe reach similar kinds of conclusions, or maybe even we'll discover that they have a chemical network that looks like life. The problem is, what are we likely to really bring back from Mars? We're really going to bring back these crazy vulcanized rubber compounds that I showed you from before that were seen in the Curiosity rover. We're going to bring back refractory carbon grains. We're going to bring back kerogen that looks like this snarled knot of carbon atoms. How are we going to work on that? And this is a huge challenge. And as I would say, it's the point of the spear and what my group is doing. Here's an illustration of something that we did. And if that's like somebody trying to escape, you can tell them, like, don't try so hard. I'm almost done. So this is
please take your time. The janitor was.
Okay, well, this is for the janitor too then. So this is the molecule lignin, which is, it's not kerogen, it's not this macromolecular material, but it's something life makes as a structural component of leaves that's just complicated enough we feel like we can kind of work with it and learn how to work systematically with these complex macromolecular things. Lignin is full of these little groups called methoxy groups. They're a methyl group, CH3, stuck to an oxygen atom that's just part of the structure. So when lignin gets buried and heated and is on its way to turning into this refractory goop that we find in the geological record, it loses the methoxy groups. That's the main thing that happens, is they get chemically cleaved off, and then the remaining atomic site cross-links with somebody else. And so this chemical procedure, it's some very specific thing that if we could understand it, maybe we could then back out what it was like before it underwent that process. So my group figured out a way to characterize the isotopic contents of these methoxy groups and observe how they change as the methoxy group leaves the molecule, or as the organic matter matures through this sort of empirical measure of its structural compactness, okay, of its folding up into this little kerogen-like kernel. As it undergoes that process, we're distilling off carbon-12 and enriching the methoxy groups in carbon-13 with a very specific pattern. That is just one process of many that matters, but it shows you we have a toehold on this question. What do you do with these sorts of materials? Our intention next is that we will take these kerogenous materials and cook out of them polycyclic aromatic hydrocarbons that we can study as our main sample of what the structures of these things are like. And the last science point I'll leave you with, here we did this on polycyclic aromatic hydrocarbons from primitive meteorites. And what we discovered is that the doubly substituted version, the version with 2 C13s in it, is radically fractionated by like over 4%, which for us is a lot, between different isomers of the same stoichiometry. So these differ only in the sort of stereochemistry of how they got assembled. There's the same number of carbon atoms, same number of hydrogen atoms, probably they came from the same sources, radically different probability that 2 C13s ended up in the same molecule. This sort of chemical isotope effect is very, very subtle under normal conditions. Way, way less than 1% would be typical. Here it's over 4%. This is orders of magnitude stronger than is typical. What we're conjecturing is that this is a signature of molecular cloud ice grain chemistry. And the reason is that at temperatures of where disks and planetary bodies and stellar envelopes, at all those temperatures, the chemical isotope effects of 2 rare isotopes touching are like negligible. At 10 Kelvin, they're big, okay? They're multiple percent. And so this is just a finger grip on what must be a complicated problem, but we think that this is likely a signature that we're only going to find in complex organic matter that gets synthesized in interstellar settings. Okay, that's it. I'll leave you with just a couple summary statements. So we have figured out how to do something that seems kind of special and lets us reconstruct chemical reaction networks, not just in the lab, but in nature. We have a kind of existence proof of a cyanide-dominated chemistry that will get you part of the way towards the chemistry of life. And then really our direction in the future is to figure out how to work with these complex macromolecular materials that we think are most likely what is going to come home from Mars. And I'll stop there.
Well, that was really remarkable. Well, first of all, let me thank you. It was so pedagogical for those of us who are not, haven't followed this in any detail. But it's quite phenomenal that you can sort of see what the history would have been of the construction of these things. Well, let me ask for questions from the audience and here.
I have a question.
Shura has a question. How do we do this?
I do not know.
I'll just listen. I'm listening carefully to you.
You are able to hear?
Yeah, I hear you just great.
Oh, good. So I apologize. My question may be vague and too complex, but I will try.
I love it.
I will try anyway. So it's very beautiful, this reconstruction of what could have happened in meteorite and how simple organic molecules could have been synthesized. My feeling, however, is that this method fundamentally brings you only up to the place where life starts.
Yeah.
And this is a very fundamental thing, because your method is based on the assumption that every molecule is generated, is synthesized in a number of copies, such that all combinations of isotopes are statistically simple.
Okay, almost. So the premise is we are looking for deviations from that statistical behavior.
Yes, but in order to see that, you have to assume that there is statistical behavior in the first place. While in any biological system, what happens is vast majority of combinatorial possibilities are never realized, not a single time.
I push back on you with this. So first of all, we're not the idea of a random distribution is a concept that we use for reporting data sometimes. Like we will say, this version of a molecule is more abundant than it would be if it was random, or less abundant than it would be if it was random. But there isn't an expectation that a particular process will follow a random rule. Second point, the propensity of life to do things that are not statistical, that's maybe true of like enzymatically controlled stereochemical reactions. But what we're looking at is difference. Let's imagine every molecule of some precursor is forced by the factors you're referring to, forced by enzymatic selectivity to undergo a certain reaction. But that molecule isn't just that molecule. It is thousands of different versions of that molecule. And each version has a different probability of undergoing that reaction in a unit time. They're competing with each other in a way. And the enzyme doesn't really know how to select. It's not selecting just one. All of them are getting through at some rate, but they are being fractionated from each other by it. So, and that then imparts a signature.
Well, I agree, and this is why I said that my question is vague, and maybe I should.
No, it's okay. There's also
I should think more about how to formulate this question.
There's a great question behind your question. So behind your question, I would say is, okay, wonderful, or maybe you like it, maybe you didn't. I think it's wonderful that we were able to reconstruct a kind of
I agree, it's
that happened in the lab.
beautiful.
Okay, But
Gorgeous.
But now, how can I tell? Imagine now somebody sneaks in the middle of the night and they take out all of the non-biological amino acids from my sample and they smear it with an apple peel or something. They put the amino acids of life in. How would I know? How would I recognize life as different from non-life? And there's like a crude answer that's just forensic matching. If I notice isotopic patterns that I have seen in terrestrial life, well, then of course it's just a contaminant. But what if it's life that's not from Earth and it's doing something different? Now you have a very deep problem. What are the characteristics of a reaction network that we would accept as evidence of life? And that's not for me to decide by myself. Basically, my job is take all these molecules and tell you how they relate to each other. That one came from that one, came from that one, came from that one, which also made that one. These sorts of things I can map out with the isotopes. Why it occurred, that's then a question, like a deeper chemistry question. My feeling is that if this line of evidence leads to something that's like an argument for life, it will be that we reconstruct a reaction network that is so idiosyncratic and weird that it can't occur in the lab without the help of enzymes.
Okay, I better stop.
Paul can ask his question while you think, reformulate yours. Yeah,
I
Paul?
can ask, you said I can ask my question?
Yeah.
Okay, so it's really beautiful stuff. One thing is, I mean, you sort of centered on life that would be like life on Earth with the chemistry we use here. But I assume what you're thinking is that if you find some molecules of some non-organic stuff somewhere, you could also look for the signature of different, what would be made naturally, let's say, from something that's different from it. And you'd interpret that as there being life of a different form. Is that sort of right?
So there's two sort of versions of the question that you're asking that you will find represented in the history of the development of this problem. One of them is a kind of extreme wide view of what life could be. Life could be made of silicon, life could be made of germanium, life could, like, all kinds of wild ideas. This was a popular sort of armchair philosophizing that occurred in the 70s and 80s, I would say, and now has kind of disappeared from this field. And I think that people basically, because they didn't feel like there was really any empirical evidence that you could start with exotic non-carbon-based chemistries and get to life, and then you look up in the sky and it's like the carbon-based chemistry of life is literally everywhere. So, like, let's just take the simple answer, like take the win that this is probably the direction we should go. And the community as a whole has basically abandoned exotic materials as the substrate of life. The second version, though, is very much alive and well, and that is to use isotopic abundances and just inter-element proportions of metals that life uses as metabolites, like iron, okay? Life likes to oxidize iron or reduce iron as a way of harvesting energy. And to use that as an indirect signature of life. And there's a whole school of work on that. Personally, I'm not compelled by it. And the reason is that just as all of the chemistry that life does to fractionate isotopes of carbon and hydrogen and nitrogen, that's just chemistry that life did. If life wasn't there, chemistry also fractionates the isotopes in very similar ways. If all I have is one metal, like if all I know is iron, that's not enough information for me to figure out the mechanism by which it was oxidized or reduced. All I know is that it happened. And so I'm motivated to steer into a type of observation where my composition space is not a line, where I move to the left or I move to the right. My composition space is 100 independent dimensions. And life moves me in a very specific way in that dimension. And all these other processes move me in different ways.
Okay, now another question, which is much easier, but I don't know the answer, which is if you, first of all, the carbon-13 concentration depends on temperature, right, for the different reactions.
Temperature can impact the amplitudes of these isotope effects
Right.
in a couple of different ways.
And if you take the products, the same product from different species, from Archaea and what, it's the same? The ratio?
Okay, yeah,
Yeah.
this is a great question. What is the temperature signature on elementary chemical processes that life is doing? There are pretty well hashed out answers to that for chemical processes that life does that are basically reversible or nearly reversible inorganic chemistry that life has harnessed. So like shelly fossils precipitate carbonate minerals. That's what the shells are. And it's life did it. I mean, it's biochemistry, but the elementary reactions of taking a carbonate ion group and sticking it to a metal and growing a crystal, that also happens without life. Those reactions have very well-known temperature dependencies, and we use them as thermometers. In fact, this is how I did the temperatures of the body temperatures of the dinosaurs.
Very cool.
That's how it
Okay.
worked.
Yeah.
But other reactions, like the enzymatic chemistry that allows the citric acid cycle to work, nobody really knows what the temperature effect will be on that. So that is something that eventually either me or somebody who does measurements of the type that I do will have to unpack.
And pretty much everything uses the same enzymes. Every species uses the same enzymes.
No, some are highly conserved, some are unique to individual species, and everything in between. So like the tree of life is composed by looking at structural variants of a highly conserved nucleic acid that is a template for making a protein folding protein. So basically everything has to fold proteins. If you're it sounds like a paradox, but in order to fold proteins, you need other proteins. And so the proteins that fold proteins, everybody has them, and slightly different versions of them. And that's how they go from, a bacterium to an oak tree to you in one continuous set of observations.
Okay, thanks. Great.
Did you say that all these different living things have one common protein folding protein?
There's a whole bunch of things that are quite highly conserved. I'm not an expert enough to say like every single living thing has exactly some version of the same protein, but it's almost like that.
Oh, here's a question in the room. Can you speak up?
Hi, I had a question. It was kind of from the center of your talk. I was just really interested in, and you might have mentioned it, about the centrality of cyanide in especially like the lake hypothesis. It was just interesting that was such a central part of it. Could you elaborate on that?
Yeah, and the fact that cyanide is important in the chemistry that we see on the meteorites is it ends up being super important and very restrictive to the natural settings where that chemistry could occur. And the reason is that cyanide is generated photochemically in the interstellar medium and in disks and nebulae and things like that. It's not hard to make cyanide in the vapor state where it's cold and there isn't too much it can bump into and react with. But cyanide in even moderate conditions, like the conditions of the Earth's surface, is very unstable with respect to oxidation. If it's in water or if it sees anything, it's almost like too reactive. So you can't really sustain a cyanide chemistry in the Earth's surface environment unless you feed it, or it is episodic with little pulses of resupply. Cyanide doesn't come from volcanoes, it only gets generated in the Earth in any abundance through atmospheric reactions, but then it doesn't persist. And so the folks who want to have the origin of life on the Earth, or this is also part of the reason why they landed it in the crater where they landed, where the Perseverance rover landed, they landed in a lake because they're thinking life emerged in a lake. So if you're going to do it there, your biggest problem is getting cyanide in and storing it. And so one of the things people think, there was just recently discovered a new form of cyanide in meteorites, where it forms as an iron COCN complex that is quite stable. And so people are imagining that you make complexes like this and then episodically leach them or break them down thermally or something to release them so they can do their job before they dissipate.
That's so interesting.
Thanks for asking that. I was too shy to ask.
Yeah, and it's a huge problem because basically all, like, if you can't make a carbon-carbon bond, you're done. Like you can't be alive if you're not able to make carbon-carbon bonds. And making a carbon-carbon bond without cyanide, you can do it, but it ends up making very narrow ranges of chemical moieties. Like the envelopes around carbon-rich stars, sure, they make lots of carbon-carbon bonds. They make acetylene and all kinds of stuff. They don't make anything useful. All they make is like very reduced soot, and that's it. And that's true also in hydrothermal vents, also in the lab. If you don't have something like cyanide around, you're in big trouble.
Well, give people one more chance to ask questions. And remember, he's going to come and visit us in the
Yes,
fall.
we'll have a blast. So now that you all know me, I was not willing to give my seminar on our failed dark matter search because I would sound like a jackass talking about it for an hour. But now that you've seen me, I'm willing to do it. So I'll give a seminar on that. I'll do a dinosaur thing and a ridiculous person looking for dark matter thing when I visit.
Very good. And so you'll come for the whole semester, right?
I don't know about that. I'll come for as many nights as the restaurants in New York can sustain.
Oh, so a semester, several years. Okay, great. John, thank you so much. Really.
Thanks so much. This was really fun.
Bye-bye.
Yeah. Easy.
Great.
Thank you, pleasure. It was very nice.